Today, I would like to start with a question. How do we improve our ability to understand biological complex phenomena like cancer, especially in humans? Let's hear what this humble petri dish has to say. A petri dish is a transparent dish which biologists use to cultivate cells at will, and it is telling to us, hey, there's a big difference between a flat layer of cells and complex three-dimensional tissue. But what will it mean? Every patient is different, and thus every cancer, which exhibit various mutations, different microenvironment, and also different responses to drugs. Traditionally, to study the complexity of cancer, appropriate model systems are necessary. So far, in vitro 2D cell culture and mouse models have been considered the pillars of preclinical evaluation of different new therapeutics in all branches of biomedical research, but they still suffer from some limitations. 2D cell culture is disadvantaged because due to its culture condition on a plastic substrate, and it is unable to mimic in vivo situation. On the other hand, mouse models are time and cost consuming, and one might argue that do not generalize perfectly patients. And finally, human tissue has a limited availability, and it is difficult to manipulate. But if we want to understand cancer, we must be able to create its environment and the cancer itself. Let's imagine to biopsy a tumor and to grow it in a lab. We are not in Alice in Wonderland, but it is already started to begin, and it is happening in many laboratories worldwide. It is considered a breakthrough for cancer treatment, and I'm talking about mini organs in a dish or organoids. Organoids are a group of cells in laboratories that you can grow in three-dimensional structures. They contain multiple cellular types of the in vivo counterpart, cell organized similarly to the primary tissue, and work similarly to the parental organ. Organoids can be derived from just small fragments of every tissue of our body, like intestine, pancreas, and lung, and many others. But researchers have also started to grow organoids from cancer patients, from human cancer patients. And this is exactly what I'm doing now for my PhD. We take biopsies from colorectal cancer patients. We isolate tumor stem cell from the bad tumor, and we immediately embed them in a scaffold that we will call extracellular matrix. And then, like this, you can be able to expand them and to in a three-dimensional structure. And finally, we want to assure if the patient-derived organoids resemble both genetically and histologically the primary tumor. So, organoids can be considered as a bridge between 2D in vitro models and in vivo models, but they have also potential in clinical aspect. Not only for cancer modeling, but also for biobanking, regenerative medicine, and to test efficacy and toxicity of drugs, and for precision medicine. We are still at the beginning of this new revolution, but with time we will get a more accurate model for resembling human physiology. Thank you very much for your attention.